All right. When we're doing this lab, okay, that we're going to do on Wednesday, you're going to be doing a couple of different tests, okay? The first test, and probably the one that's going to give you the most information, okay, is what we call the Observable Properties Test, okay? So that's this. That's the first thing that you're going to do. And I'm telling you this because tomorrow you're going to be designing your own procedure. You need to know what tests you're going to perform, okay? These are the tests you're going to perform. You just need to write up kind of how you're going to do that. All right. Observable properties are anything that you can directly observe without having to alter the sample in any way. Okay, without adding anything to the sample, okay, without reacting it with anything. It's basically just your 5 senses. Okay. Well, sorry, not your 5, 4. 4 senses. You're not using taste. Okay, we're not allowed to do that, so you're only using 4 of your senses. All right, so one of the observable properties is color. And we've already said that if you see pink, red, okay, green, or blue, you should already kind of know the possible, one of the possible identities for those compounds. So color could be important. Okay, state of matter is something else you're going to want to observe and record. Is it a solid? Is it a liquid? Obviously, we just said there's going to be no gases, okay, but state of matter is going to be an important one. Okay. The nature of the matter. If it's a solid, is it crystalline? Is it waxy? Okay. Is it cloudy? Okay. Things like that. So we need to know, okay, what's the nature of this matter? Okay. If we're talking about a liquid, is the liquid clear? Is it cloudy? Okay. Is it kind of a colloid like Play-Doh? Okay. Then it's kind of semi-solid, right? Things like that. Okay. Another thing you would probably want to do during an observable properties test is check for odors. Okay, so to do that, you would do wafting. All right, I'm going to let you copy that down here. I'm just going to go grab a couple things from the lab so you can see what I'm talking about. Okay, so some of the stuff you're going to be using on Wednesday. Okay, this is what your depression plate will look like, pretty much like I described it, right? It's got little pits in it. Okay, it's not called a depression plate because it's sad, right? It's got little depressions in it. Okay, then that's where your samples are going to go. Now, for some of these observable properties tests, you're probably not going to want to do the observable properties test when you've got all of them side by side in this little dish, right? Because if you're trying to do a wafting test, you're probably getting all of them at the same time. Agreed? Okay, so you might as well do the observable properties test by walking up and looking at the larger samples that are going to be at the front. Okay, especially for the wafting test. All right, you're going to want to now. I can save you some time and tell you none of the solids have scent. You don't need to bother smelling any of the solids. None of them have any smell. Okay, but you'll probably want to smell the liquids for sure. Okay, so we'll do the wafting test. Okay, nature of the matter. Okay, color. Obviously, we talked about that already. So those are the things you have to do them first. Okay, because you can't do them after you've done any of these other tests. So there is a specific order in which you need to do the test because each test changes your samples just a little bit, and then it would make it impossible to do a test before that. Okay. All right. So we've looked at the observable properties. Okay. And when you're doing these tests, you do need to make a chart or something. Okay. And I'll kind of show you how that's going to work, where you can record all of these observations. And you're going to want to be pretty thorough. Okay. I've seen people do this lab, and they write things like solid, blue, good, bad, 7. Okay, and that's their like, I'm like, I couldn't identify that stuff from those observations. How in the heck are you going to do it? Okay, so you need to make really thorough observations. Really thorough observations of something like copper sulfate, okay, would be, okay, blue crystalline solid, shard-like crystals. Okay, so not square, like kind of pointy. Okay. So that kind of covers those two things. Odor test, no odor was detected, and then I could move on from there. All right. Whereas, you know, just telling me things with one word isn't really going to be useful later. All right. The next test you're going to want to do is your solubility test. Okay. Solubility test is pretty straightforward. You're going to add distilled water to all of your samples. All right. So when we're doing this on Wednesday, you're going to fill these little depressions about 1/3 of the way with the unknown sample. All right? And then when you do the solubility test, you'll fill them so that they're about 3/4 full, but no more than that, okay, with water. right? And then you can stir them around with a little stirring rod, a glass stirring rod, okay, in order to see if there's any dissolving. Now, just because all of the sample doesn't dissolve doesn't mean that the sample wasn't soluble. Okay? If I had blue crystals and now the water is blue, did it dissolve? Sure it did. Okay, I have evidence of that. Okay, or if the crystals look smaller, or it looks like there's less of them, then it's dissolving. Okay, all of those are signs that yes, it has dissolved. Because in this much space, you're probably not going to get that amount of stuff to dissolve, but you'll get some of it to dissolve for sure. Okay, so those are kind of things you're going to watch for, okay, with the solubility test. So you're going to see, does it dissolve? And at the same time, you're going to put the thermometer in there and see if there's any temperature change. Okay, when you add acid to water, it generally gets hotter. Okay, in fact, when you're diluting acids, you always add acid to water and not the other way around. Okay? If you add water to a concentrated acid, it gets so hot that it can actually break the glass that it's in because the glass can't expand fast enough. Okay? And so it'll shatter. So you always add acid to water to help keep that temperature change down a bit. Okay? So we'll look for temperature change, color change, and if any new odors are produced. Hopefully that doesn't happen. Okay? Okay, questions on the solubility test. Okay, so we do this one first, observable properties. Then we do solubility. Okay, then we'll do the conductivity test. Because obviously we can't test the conductivity of a solution until we have one. So it makes sense to do it after you've done the solubility test and now everything is dissolved in water. Alright, so the description there is you're going to look for the little lights. So your conductivity testers, most of them look like this. We have a few other ones that have a slightly different design, but they're still generally the same. Okay, there's a little button on the side here. Okay, you press that button and the on light comes on. These are vastly superior to the ones we used to have, which did not have an on light. And sometimes the battery was just dead, and then everyone said there was no conductivity for everything. Well, that wasn't very good. Okay, these ones let us know if at least the battery's working. Okay, so you press the battery, or you press, the button, and you stick it in the solution. And depending on the conductivity of the solution, okay, more lights will light up. All right. So generally, if you've got something that's got a conductivity of 6 or greater, you've got an ionic compound. Anything that's like 5 or less, and you're looking at molecular. Most of the time, you're going to get like 8, 9, 10. Or 1, 2, 3. You don't generally get too many in the middle. Okay, they're either obviously ionic or obviously molecular. Okay, everyone with me on that? Okay, so that's an easy test, and really, your only observations that you need for that test is the number of lights that lit up. Okay, or you could say highly conductive or non-conductive. Doesn't really matter either way. Okay, you need to make sure you rinse this in between. Right? Otherwise, you might transfer ions from one sample to a sample that's molecular, and then you'll get false readings. So you need to rinse this. But do you want to rinse it with tap water or distilled water? Distilled water. If you rinse it with tap water, you're rinsing it with ions, okay? And that's not going to help your situation. So you always rinse it with distilled water. We'll have little wash bottles, okay? They have a squirt top on them, and you can just squirt them off, okay? So that test is, again, relatively straightforward. You just have to make sure you rinse it in between steps. Okay, once we've done the conductivity test, then you can do a pH test. All right, pH test will simply involve taking some Hydrion paper, okay, which is this stuff here. Okay, you've probably used that before. All right, you just break off a really short piece for each sample, maybe about a ½ an inch long. So let's say about like that. Okay, just dip it in the solution and compare it to the color chart. Okay, and that'll be your pH reading. We have to do this test last, okay, because a color change is an indication of a chemical reaction. Once we've done this test, we have chemically altered our sample, okay, and it is no longer what we started with. That's why the pH test has to be done last. Okay, now I've written here very, very brief descriptions of how to do these tests. Tomorrow, you and your group are going to be writing up your procedure. Should your descriptions of the test be much more thorough? Yes, they should. Okay, you have to write a procedure like you're expecting, like the dumbest person in the world to try and do your lab. Okay, if you take anything, if you make any assumptions that they know what they're doing, they'll screw it up. Okay, so it has to be written like a cookbook. You do this step with this much of this stuff, and you look for these things and write it down. Okay, and then step 2. You do this test by doing this and this and this. You look for these things and you write them down, and then you do this. So you got to make sure it's very, very thorough. Okay, very, very explanatory on how to do each and every step. All right, everyone good with that? Okay. Now, in terms of lab layouts, okay, what you're going to get in Google Classroom, okay, from me is this exact file. Okay, for this lab. So it's a template. It's already partly done for you. Okay, so it'll say problem. Okay, restate the problem in your own words. So on the lab sheet that I give you tomorrow, okay, will be the problem that we're investigating. So you need to read that, think about it for yourself, and say, okay, the problem is kind of saying this, and write that down. Just don't copy and paste my problem, because that's not going to get you any marks. Okay, so restate it in your own words. Then you've got your lab design, where you identify your manipulated variable, your responding variable, and your controlled variables. Okay, so you'll identify them and you'll explain. This is the manipulated variable because, and we're going to go over the variables again here in a few minutes just so you're not lost. Okay, this is the responding variable because, these are the controlled variables because. Okay, then you'll have your hypothesis, which is always an if, and then statement. Okay, again, we'll talk about that as well here in a few minutes. And then you've got your materials. Okay, this also contains all of your unknowns. From there, you'll have this chart. Okay, so I've already put the observations chart in your lab report for you. Okay, all you have to do is put in your observations. Okay, and if it needs to expand to be more lines than one line, which it probably does, it'll do that automatically. Okay, and you'll be good to go on there. All right, so again, the compounds will just be labeled A through K. Okay, that'll be your data and observations, and then you'll have your analysis. Okay, you've got to decide, was sample A ionic or molecular, and which of these things was it? All right, so if you thought sample A was ammonium nitrate, you would say, okay, it was ionic and it was ammonium nitrate. All right, everyone with me there? All right, so that's what you're going to be doing for that part. Then you'll have, okay, then you'll have your conclusion, and in the evaluation you identify any sources of error. Okay, anything that you thought we could have done differently, okay, to make the lab easier or more valid. Okay, you put those in there. So you say, this I thought didn't work very well. Here's how I would fix it. And you got to usually identify at least 2 of those. Okay, and then there's class discussion. Those are not questions that you answer. They're questions that we'll go over as a class after I hand back the lab report. Okay, and then below that is where I'll give you your marks. Okay. Everyone follow me there? So that's the file that you're going to get. Okay, all you have to do is fill in the stuff. Okay, the template is already built for you. All right, so tomorrow you'll have that, and you'll start. Okay, you'll start filling in the procedure here. So you'll fill in like step 1. Go get the materials you need. Okay, that would be, and then make sure you say, go and get a depression plate and this and that. All right, step 2, perform this test. Here's how you do it. All right, making sense? All right, so problem is always going to be stated as a question. Right? I don't always state it as a question on the sheet that I give you, but you always do. All right, so when you're writing out your problem, okay, it's like Jeopardy. Right? You must state your answer in the form of a question. You must state your problem in the form of a question. All right, so an example could be, what is the effect of light on plant growth? All right, that could be a problem that someone was investigating if they've been living under a rock their whole life and didn't know what effect light had on plant growth. Okay, just to keep you from writing the whole thing down all at once here. Okay, the next part of your lab report is the design. Okay, and then in the design you decide what What am I going to manipulate? What am I going to change in this lab, me as the experimenter? Okay? What am I going to be watching for? Okay? That's your responding variable. Okay? And what kind of things do I need to make sure are the same for all the trials that I do or for all the subjects or whatever? All right? So the example here that we have is, what's the effect of light on plant growth? So light, then, is probably going to be my manipulated variable. All right? It's the thing that I'm going to decide how much light do the plants get. Okay? I would say a lab to test this would probably involve several plants. Okay? I would have a plant that would get, let's say, 24 hours of sunshine. Okay? Maybe I'd have another plant that gets 18 hours of sunshine, one that gets 12 hours of sunshine, one that gets 8, 4, and one that's in total darkness all the time. Okay? That gives me a pretty good spectrum of exposure to light. Agreed? Okay? So I'm manipulating the amount of light the plants are going to get. What am I watching for? Their growth. Okay? The problem says, what's the effect of light on plant growth? The problem almost always tells you the manipulated and the responding variables. Okay? What's the effect of this, your manipulated variable, on this, your responding variable? All right? So I would have to be measuring then the growth of plants. And I might measure that in a couple of different ways. I could say the responding variable is the growth of the plant, measured by, let's say, number of leaves, height, and mass. Maybe I would use 3 indicators of the growth of the plant. Okay? I could also have a qualitative thing where I could just say plant's appearance. If the plant looks sick, it's probably not doing so well. All right? Those could be qualitative observations as well. Okay? Now, are there other things that affect the growth of a plant? Right? Water affects the growth of a plant. Soil affects the growth of a plant. The plant itself, like, if I'm going to have all these different plants getting different amounts of light, should they all be the same kind of plant? Yeah, I can't compare, literally here, apples and oranges. I can't have an apple tree, an orange tree, a pine tree, and then like a little ficus. Okay? That's not a fair comparison. That's what we call an uncontrolled experiment. I have not controlled all of the variables that could affect my results. My results are the growth of the plant. So my controlled variables are the things I know could affect the growth of a plant, so I'm keeping them the same for everybody. Okay, so those things would have to be, all the plants are going to get this much water. All the plants are going to be in a room that is 20 degrees Celsius. All the plants are getting potting soil I bought at Walmart in this bag, this brand, so that's all the same. Okay. Maybe if I would choose to, I can make sure they all get fertilizer, but I got to get the same fertilizer in the same amount. Okay, all of those things that could otherwise affect my results are my controlled variables. Right? So when I list them, I say all plants are getting the same amount of water because plants require water for growth, and if it's not the same, they'll have different rates of growth. All plants will get the same amount, or will be in a same temperature room because temperature can affect the rate of growth of a plant. Okay, things like that. Does everyone follow? So when you're explaining these, you need to say, it would affect the experiment this way if I didn't keep it the same for all of them. Okay, so the manipulated variable here is the part of the experiment that's changed on purpose by me, the experimenter. Right? And for this one here that we just talked about, that's the light given to the plants. Okay, and then a responding variable, okay, is the thing that, okay, we're watching for. Well, hang on here, guys. That should be there. I don't know why that moved, but okay. So the responding variable is whatever changes as a result of the changes you made. So as a result of the manipulated variable. Okay, so it's in this case going to be the growth of the plants. All right, and then we said the controlled variables are the other things that can affect the growth of a plant. And we want to keep them the same so that they don't affect the growth of the plant. Okay, or at least they affect the growth of the plant exactly the same for all of our plants. Okay, and these are usually environmental factors. Okay, controlled variables are almost always environmental factors. Okay, so let's look at a different example, maybe not plant growth. Okay, I'll give you one from when I was in university. A bunch of buddies of mine and I, we got into this study that someone in the phys ed department was doing for their PhD. They were testing the effect of creatine on muscle growth. Okay, and they needed subjects for their experiment. Okay, so we all got these containers, okay, that were filled with essentially a shake, okay, and we had to make sure that we took this shake twice a day, and then they gave us a workout program that we all had to follow, right, for like the next month. Right, so we all did this over the course of the month. We did the workouts that we were supposed to do, and we did them. All of us did them at the same time. Okay, and we ate the shakes at the same time, same volume of shakes, all that kind of stuff. Okay, and at the end of the month, what they did was at the beginning of the experiment, first they had us do just strength tests. Okay, they weighed us. They did some like bench press, squats, things like that to see what our maxes were, and then after a month we did the same tests, okay, so that they could see if there was any improvement, any gain in weight, okay, and all that kind of stuff. All right, now the controlled variables for that experiment were that we were all kind of university age students, okay, we were all male. Okay, I'm sure they had a female group, but we didn't know because we weren't part of it. Okay, similar physical characteristics, and then we all did the same workouts. Okay, we had very similar diets. At least we all had the same shake all the time. Since we lived in the same dorm, we were eating the same food all the time as well, so it worked out great for them. Okay, we worked out at the same time. We did the same workouts. All these things were the controlled variables. Because if one of us went and worked out 3 times a day, and the other guy sat in front of the TV for 24 hours, okay, it's not really going to give them really good data. Okay, so they had to make sure that they controlled all the variables, okay, that could affect muscle growth for people. Okay, now ended up that not all of us actually got the creatine. Okay, they gave us all a container that contained a shake, but not all of the shakes contained creatine. Why? Right. they have to have what's called the experimental group. That's the people that are actually being experimented on, okay? And what's called a control group, which is your comparison group, okay? And unfortunately for this guy, what he discovered was there was actually no significant difference, okay, between the people who got the creatine and the people who didn't. All right? So in the end, in terms of strength, there was no significant difference in gain, but there was significant difference in gain in weight. Right? Because what he ended up finding later was that creatine allowed your muscles to absorb more water and thus appear larger, even though you weren't really gaining a lot of strength difference, okay, between it. But you would look bigger because the creatine allowed your muscles to essentially swell, okay, which would be useful if you were in a bodybuilding competition and wanted to look giant, okay? Everyone kind of follow me there? All right. Now, what was his manipulated variable for that experiment? Right, whether you got the creatine or not. That was the manipulated variable. Okay, the responding variable was muscle growth. Okay, there was actually several responding variables. There was mass, how heavy we were before and after. Okay, and difference in strength. Okay, so did we make any? Was there any difference between the people who got creatine versus not creatine in terms of how much gain they got in terms of their strength? So there were a couple of responding variables. Sometimes experiments are like that. Okay. And then the controlled variables we've already discussed. So does that sort of make sense? Okay. That's kind of the way it works when any pharmaceutical company is testing a new drug. When they get to the point where they're doing human tests, okay, they'll have what they call the experimental group. That's the people that actually get the drug. And then they'll have the placebo group. Okay. A placebo is a sugar pill. Right. So essentially, you take a pill just like everybody else, except your pill has got nothing in it but sugar. Right? So you're not taking anything. So if they're testing, let's say, a new allergy medication to see if people get relief of their allergies, right? If the people who take the medication and the people who don't take the medication feel the same, then their drug doesn't work. All right? So if the people who took the placebo felt better and the people who took the pill felt better, then the only thing that worked was taking a pill. Okay? And you could take anything you wanted and it works. Right? And there is actually studies on something called the placebo effect. Okay, mind over matter. Simply taking a pill and saying this pill is going to do this, people will go, oh, okay, I feel better now. Okay, just because they took a pill. Right? They actually used to do that in like the Korean War and World War II. If they ran out of morphine, okay, they'd give the soldiers a placebo and say, here's some morphine. Right? And oh, okay, oh, I feel better, even though it wasn't really anything. Okay, it's just their body told them they felt better. All right, everyone getting the variables here? Okay. Now, probably the most important part of your experiment is the hypothesis. Okay, sorry, we listed these controlled variables before. I'm just going to skip over those. Okay. Your hypothesis always contains these three words: if, and, then. Okay, because it's a deductive statement. Okay. A lot of people have this idea that a hypothesis is a guess, and it's not. Okay, it's not a guess. It's not the answer so much to the problem either. Okay, what you're saying in a hypothesis is this: if this is true and this is done, then this is going to happen. Okay, it's a prediction. All right, it says, I think this is the cause of the problem, or I think that this is what's going on, and this is what I'm going to do to test it. Then this is going to happen if I'm right. Okay, so if I'm doing this experiment here on plants, okay, and light on plants, then I'm going to say something like this. Okay, if light positively affects plant growth, and plants are given differing amounts of light, then the plants getting the most light will show the most growth. Okay, is that a reasonable hypothesis? Okay, it says what I think is the solution. Okay, it says I think the more light you get, the more you're going to grow. Okay, I tell them briefly how I'm going to run that experiment. Okay, I don't go through the whole procedure. I just briefly describe it. I say some plants are going to get more lights than others. Okay, then the plants that get the most light are going to grow the most. In a hypothesis, you have to set yourself up to be either right or wrong. You cannot sit on the fence in a hypothesis, or you'll never be able to have a conclusion. Okay? You have to either be right or wrong at the end. Everyone follow me there? Because I have a lot of people whose hypothesis would go, if light affects plants and I give plants different amount of growth, then plants will grow better. Which plants will grow better? Okay, how am I going to answer that at the end when I go to my conclusion? I said plants would grow better. Some did, some didn't. I don't know. Okay, you have to say this is going to be the results. That way, if those results don't show up, you can say my hypothesis is wrong. I reject it. Here's an area where I might change things for further research. All right, or you find unequivocally you were right. Hey, all the plants that got 24 hours of light growed like crazy, and the ones that grew like crazy. Okay, and the ones that were in total darkness died. I was right. Okay, well, you should never say I was right in a conclusion, but you can say it is then true that light positively affects the growth of plants. Okay, so you can't sit on the fence in a hypothesis. So it's an if, and then statement that communicates what the answer or relationship in the problem is. The relationship in our problem is that the more light plants get, the more they're going to grow. Okay, so it's got to do three things, guys. Okay, your hypothesis always has to do three things. That's why it always contains the words if, and then. The if part says if this is true. The and part describes what you're going to do in the experiment. And the then part says your expected results. If you don't get those expected results as your data, then you can't accept your hypothesis in your conclusion. All right, okay, and then after that you'll have, okay, sorry, this was the example, okay, this was the example hypothesis there, okay, that we kind of went over already. So if plants require light for growth and 5 identical plants are given different amounts of light, then the plants that receive the most light will show the most growth. Okay. Okay, so the if part, like we said here, is the part that answers, okay, the cause of the problem or tells us what the relationship is. And you've already got that up there. You don't need to rewrite that. Okay, the and part is a brief description of the experiment. The then part says, here's my expected results. Okay, does anyone need that example hypothesis or no? All right, and we already talked about that, so we'll skip past that here. Okay, for the materials, for you guys, since I'm giving you the template, the materials are always going to be on there. So you don't even need to really write anything because they're already going to be on your lab report. Okay, I've got on here C sheet because that's what I used to have people write when they actually had to type the thing out themselves. You're not going to have to type anything for materials because I'm just leaving them on there. Okay, so it's going to save you a little bit of work there. All right, then your procedure. Okay. So it's a detailed, step-by-step set of directions for preparing and performing the experiment. Okay? And again, you got to write that as if the person performing your experiment is a total idiot. Okay? If you make any assumptions, they'll screw it up. So make everything explicit. Okay? Explicitly state you do this, you use this much, then you do this, then you clean this by doing this, and it's just, yeah, it's got to be really, really detailed. Okay, for observations. Observations come in a variety of forms depending on the kind of experiment. Right? For the experiment we're doing on Wednesday, it's basically going to be a chart filled with your qualitative observations of the color changed, your quantitative observations of the conductivity was 7 or the pH was 2 or things like that. Okay? So your you're going to write in different kinds of things. Sometimes your observations could be measurements. All right, like if we're doing a physics lab, how far did this thing travel? How fast did it go? All right, it all depends on the type of experiment. For a biology lab, it could be lab drawings, okay, of something that you saw through the microscope, or pictures that you took of what you saw through the microscope. Okay, so charts, graphs, or any other kinds of data. Calculations are usually in analysis, but you may have some in the observations. Okay, and any other anecdotal data, okay, or notes. Anecdotes means like you described a process that went on, kind of like telling a story. Like an anecdote is a short story. Okay, anecdotal notes could be, over the course of several months, I've observed that this tree has started to look more and more sick as I continue to poison it. Okay, something like that. Okay, so observation's pretty straightforward there. Then in your analysis, okay, for us, most of the time that's going to be answering questions. For the lab you're doing on Wednesday, it's going to be identifying the unknown compounds. Okay, for physics labs, it's graphs, okay, and then analyzing trends in data, okay, things like that. So analysis can be a pretty wide open type of thing. Okay, you don't have to worry about this do not recopy. Okay, you're just going to write your answers in the blanks on that template that I gave you. Sometimes I leave the questions on the template, sometimes I don't. On this one, I left them on there because it's a chart that you've got to fill in. Okay, if a question is asking you to explain something, do that. Be detailed. Okay, don't just say yes, because that's not an explanation. Right? For this lab, you're not really going to be explaining anything. You're just going to be sample A was ionic and it was ammonium chloride or whatever. Okay, things like that. Now, in your conclusion, okay, which is the last part of your experiment, you restate your hypothesis. That's going to be super easy for you. Go up to the hypothesis, copy and paste. That's all you have to do. Okay, so restate your hypothesis by copying and pasting it. Then you tell me whether you accept or reject it. Right? So in this case, if you're able to identify all the compounds, you're going to say, I can accept my hypothesis because, using the physical properties of these things, I was able to find that they were all slightly different and thus assign an identity to each one. Something like that. Okay? If we're doing the plant experiment, I'm going to say, probably my data would have said this. My data would have said the plant that gets 12 hours of light and 12 hours of dark grew the best. Okay? Because 18 and 24 hours is going to kill the plant. Okay? Less than that, going to kill the plant. Then I'm going to find that I have to actually reject my hypothesis because I said the more light they would get would be better. That's not true. It was actually found that plants need a certain amount of darkness as well. Okay? So in your conclusion, okay, restate, accept or reject based on the results. And then the evaluation, you give me your sources of error. Now the thing about sources of error, guys, the sources of error cannot be correctable human error. Okay? All the time I get this kind of stuff. Yeah, we screwed this one up. So yeah, that's a source of error. We wouldn't screw it up next time. If you screw it up, do it again. Okay? Basically, you're telling me I messed up, quit error, and I was too freaking lazy to do it again. Okay? So my results are all off. My experiment is completely invalid, but it's okay. No, it's not okay. Okay? If you list in there, I screwed up and didn't fix it, I'm going to go, okay, 0, 0, 0. Okay? We don't want that. All right? Correctable errors, okay, are things like, sources of error are things like, we were only given really small samples, and that made doing everything really hard. It would have been way easier to do if the samples were bigger. Okay? There's a source of error. There's a way to fix it. Okay? Those are legitimate things. You're not going to hurt my feelings. Okay? If you say, this part of the experiment was really hard, or this part could have been done better. I make it that way. Okay? That way you can find the sources of error. And they're kind of more obvious. Everyone follow me there? Okay, so tomorrow I'm going to give you the lab handout, and you're going to get in groups, and you're going to start writing up the procedure, okay, all that kind of stuff. And probably doing the hypothesis and design. You'll have half the lab report done tomorrow. And we won't even have done the lab yet. Okay, so this should not be too much homework for you in the end.